Good afternoon, everyone. My name is Gina Wen. I'm a SMID-cap biotech analyst at Barclays. It is my great pleasure to introduce our next speaker, John Leonard, President and Chief Executive Officer of Intellia.
Thank you, Gina. Let me spend a couple of minutes just introducing the company and giving you a quick overview of where we are and some of the things to bear in mind with respect to Intellia. Forward-looking statement. In a nutshell, Intellia is a CRISPR-Cas9 gene editing company. What we're building at the organization is something we call a full-spectrum genome editing company, and I'll talk a little bit about that in the comments to come, but in a nutshell, that reflects the work that we're doing in the in vivo gene editing side and our work to build world-class engineered cell therapy company as well. A key to thinking about us is that we're doing this work in a modular approach where we solve a variety of different problems, aggregate that technology, and put ourselves in a position where we can play that out as we move to various product candidates and gain efficiencies in the process. In the short time that we've been a company in Cambridge, Massachusetts, we've made a lot of progress, have very significant in vivo preclinical work that we've done. It's been published. And this serves as the basis for our work to move into the clinic with what will be our lead compound, amyloidosis of the TTR type. Shortly after that, by the end of this year, we hope to have our first ex vivo or engineered cell therapy drug candidate by the end of the year. And that's moving forward very quickly as well. We have a super management team that is able to take this into the clinic and all the way to approval, with a lot of experience from prior biotech and large pharma companies. And importantly, we have the capital necessary to move this forward very, very aggressively and are funded well into 2021. Now, just some things to bear in mind that I think are helpful when you think about Intellia. As I said, CRISPR-Cas9 gene editing serves as the basic technology that is at the core of the company. But our full-spectrum gene editing approach is one where we take that technology and apply it very, very broadly. And two general ways of thinking about that are where CRISPR itself is the therapy, and by that we mean that we deliver it directly into the body. It serves as the active therapeutic agent. It's the drug. And that's best suited for pursuing any number of different genetic diseases. On the other side, however, CRISPR-Cas9 can be used as a tool to actually create a therapy where, in this case, an engineered cell would be delivered back into the body, and that would be the so-called drug, and CRISPR itself would not be delivered into the body. So two different approaches linked by a modular platform and a lot of synergies and crosstalk that takes place, and that we think serves us well and brings a lot of efficiency and gains for our organization. By way of clinical validation, and again, this serves as the basis for our first in vivo project, which has a 2020 IND, is this work illustrated here. Our lead program on the in vivo side is pursuing TTR amyloidosis. This is a disease where an aberrant protein is produced, and the therapeutic objective is to remove that protein or prevent its production. And what you see here on the left side is the progress we've made with gene editing over the course of a year and a half of work. So from our first series of experiments with non-human primates, excuse me, we had low levels, but very exciting nonetheless, levels of editing in the liver, and through a series of progressive improvements, we've gotten to the point where we are most recently, where with very tight variability, very limited variability, and about 80% of all of the cells in the liver having the TTR loci knocked out, we've been able, on that right panel, with those two lower lines, to achieve essentially something approximating an ablation of TTR protein circulating levels. So What you're seeing is essentially a 95% reduction in circulating TTR. That, in the human setting, translates into expected therapeutic significance, and we think sets us up for what is a very, very competitive product when we get into the clinic and ultimately move to larger clinical trials. On the ex vivo engineered cell therapy approach, again, we're taking this modular approach where any number of different improvements can be reduced to practice by, in this case, modifying a lymphocyte. That begins with the first work stream here, where in contrast with CAR T cells, we're taking a T cell receptor replacement approach. And the idea here is that any number of different malignancies cannot be reached by the typical CAR T technology, but by having a TCR that's active against it, it opens up a wide set of tumors, whether liquid or solid, for ultimate therapeutic targeting. WT1, in our case, is the lead program, and again, this is something that we expect to have a development candidate for by the end of the year. That's accompanied by a second work stream, which is independent but can be linked to the TCR platform, and that is pursuing an allogeneic cell approach. And here the idea is that you can have a large source of cells waiting outside the body for whatever final application to be supplied to a patient. And in contrast with a lot of the work that we see elsewhere, what we've been doing is trying to take a very systematic approach to modifying the large number of proteins that are on the surface of cells and lymphocytes that trigger NK-mediated killing. So in contrast with knocking out MHC1 or 2, or merely the TCR that's endogenous, we've gone through a very systematic engineering effort to identify that set of changes that will confer persistence in the setting of NK-mediated killing. And that's something that we're making a lot of progress on. And then the final two categories, functional modulation or solid tumor efficacy. We're using CRISPR essentially as a discovery tool to find that set of targets and engineering changes that we can make that will permit lymphocytes bearing TCRs to enter into solid tumors, which is the broadest and largest number of tumors that exist, and do that in a way that modulates their functionality as they get there. So a lot of progress on TCR on the allo front, more to come on the functional modulation and solid tumor, and again, independent work streams that can be woven together to have any different array of potential products. In terms of pipeline and the work that we've progressed thus far, for the in vivo approach where CRISPR is the active agent, TTR amyloidosis is, again, the lead project, and we're in the late stages of research, entering into early development, working through our dose range finding work and moving into tox studies for an IND in 2020. Behind that is an insertion program where we're taking alpha-1 antitrypsin deficiency and dropping in the active gene. We've disclosed work in animals that's quite substantial, and then a variety of different knockouts behind it. And then in the ex vivo engineered cell therapy, again, AML with the WT1 target is the lead program, just a little bit behind TTR with a development candidate by the end of the year, and any number of different IO immuno-oncology programs behind it. And again, we have work that we've done on a collaborative basis with Novartis. Looking backwards, we've done a lot of work and made progress to this point. And as the year unfolds, there'll be a flow of data that speaks to the continued progress and dose range finding, ultimately tox work in non-human primates. And that's all on track, progressing well. We're moving towards supplying the GMP material for the tox work and what will be our clinical work. And we would expect to, later this year, talk a little bit more about the insertion data that moves from the murine into the non-human primate modality. And as I said, we expect to file an IND for ATTR in 2020. And the lead headline here for the ex vivo side is having development candidate by the end of the year. And we would expect to have some additional information with respect to progress on those various work streams as we move forward. So that in a nutshell gives you a sense of where we are and some of the things we're working on. And where would you like to go? You want me to join you? Okay, great.
Thank you very much for a great introduction and overview. So maybe I will start with specific program, the ATTR program. That's one of your lead programs for the in vivo program. Last year you shared some data with higher editing efficiency with improved the lipid nanoparticle cargo. So wondering if any color you can share with us regarding what you did with the LNP cargo and which component was optimized, and what was that to improve safety or delivery efficiency?
Well, I'm not going to give away all my secrets to you, as nice as you are, Gina. But here's a way of thinking about it. Mm-hmm. What one needs to achieve if you're going to get to the high levels of editing is solving sort of a kinetic problem, right? So, with LNPs, you've got this chemical delivery modality that is encoding or carrying the messenger RNA for the Cas and a guide RNA. And what happens when that gets to a cell is that mRNA has got to be translated into protein. So that takes time. And you've got this guide RNA that needs to find that protein at the end of that. So what you're solving for in the end is a kinetic problem. You want to have that guide floating around long enough so that it can find the protein as it's produced. So you want to make a lot of protein fast. And we found ways to optimize that, right? And so that succession of improvements in editing reflects work we've done with dealing with a variety of factors that influence the rate at which protein's made and the duration that the guide hangs around, as well as thinking very carefully about how to put that LNP together in the first place.
Okay, so your view actually is more the mRNA or the guide RNA modification make it.
Well, we've done a lot of chemistry on the guide, and we've been doing that for the last few years, and we've been very systematic as we think about what is the mRNA and what makes it work. So we've certainly worked on both of those things. That is the cargo, after all,
yes.
right? And with the LNP, I would characterize LNPs as benefiting from a very careful look at how you put them together at scale and manufacturing. So there's an expertise that comes with that, and I think that's something that we made a lot of progress on as well. So all of those things factor into this.
So another question is, I think you also showed the slides, the improved version, editing efficiency is about 80% and knockdown is over 95%. Yeah. But from experience with the knockdown, like say Alnylam and Ionis data, we know that 80% is already enough. So, are we overshooting? Of course, for the other indications, this is a higher editing efficiency always will be beneficial.
Yeah, so I would say enough is a function of having a positive clinical trial, right? So the objective is to have the maximal therapeutic benefit you can have. So which is different from distinguishing a treatment effect from placebo, which is what that trial was done. That's no mean feat, and I certainly think it's a great time with some of the approaches that are coming forward for patients with amyloidosis. But the central thesis is that you want to take away the protein, and we believe, and I think many in the field believe, I think even Alnylam believes that the more you take away, the greater the effect. And so what we've said is we will target very high levels of protein reduction because we think that is ultimately what's going to drive the best possible outcomes for patients.
Do we know, the side effect of permanent knockout of the ATTR genes and any concerns there? Is for the knockdown, therapy they can reverse if at some point, right?
Yeah, it's as far as the field knows, and this comes from following patients with knockdowns to varying degrees. There is no adverse effect of having low levels of this protein. There are knockout animals that have been produced for which there's no apparent clinical effect. They have normal lifespans and all that. And we look at people, who've had at least 80% reductions in these cases, and we can examine them. So, the protein's not absent, but it will be in very low levels. And I think that when you couple what we know already with the medical condition of people who are dealing with a mortal illness, it is a very easy kind of benefit-risk judgment to make.
Okay. And then, 2019, so you will also present some more data. Can you give a little bit more color, and then what timeline should we expect the data? And then, I will assume from non-human primates data, right? So
yeah.
Any more color you can give?
Yeah, sure. So I think the meaningful data at this point is very much on the non-human primate side, because that's the next step before humans. And so what are we doing? We're working through our dose range finding studies. That work is well underway. It's setting up our tox work. Interesting and I think important results will be something about the durability of the effect. And we have animals that have been treated for some time now. I think we'll be in a position to talk about that and provide more information. We've talked about that in the mirroring setting, where it looks like essentially for the life of the animal, the edit remains. So, we hope to corroborate that in larger animals, but we'll be in a position to do that later this year. More indication of the consistency of the effect and even higher doses from a safety and tolerability point of view, I think, are the relevant information. And I would expect to talk about that sometime in the second half of the year.
Are we talking about ESGCT or some medical conference?
Yeah, we haven't decided yet. And
Okay.
I'm not trying to be evasive at all, and we'll certainly tell the world before we get to that point. What we're trying to do is bring as many elements into a story and have a complete one, as opposed to dribs and drabs. So some of this is just the maturation of the data as we get it.
Okay. And then also, regarding, you also mentioned the LNP cargo, the one component you improve a lot is the guide RNA. So this is a non-human primates data. So regarding the clinical asset, right? So the one you will move to clinic, that I would assume will be human guide. Will you share with us would that be also the new assets? And then will you share with us about the off-targets data and all those, the specificity data?
Well, I won't be able to use that human guide in a monkey and get the same results, because there's a very minor sequence variation to it. But the guide selection process that led to this work is identical, right? It's just on the human cell. So, I think there'll be a time to go through that work and show the data of the human guide in human cells. It is at least as active as what we've found in the monkey. So we're very excited about that.
Okay. And the off-targets that you also showed? Yeah, sure. Okay. And then next, is the IND filing. I think a lot of the investors have been waiting and are very excited about this potential first, in vivo clinical trials. So can you walk us through the steps that require into the 2020? And then, by the way, is that first half 2020 or second half 2020?
I knew you were going to ask me that. So let me do the second part first. We're saying 2020 as the studies progress. We'll tighten that and we'll give guidance as to more definitively where in the year that sets. Everything that we've laid out in terms of timelines is absolutely on time. Things are progressing very, very well. We are comfortably in 2020, so I think we're in a great position for that. In terms of the work that needs to be done, it's really assembling the parts at scale and manufactured appropriately so that you can do the necessary toxicology studies and some of the additional dosing work that we're looking to do, and then ultimately make those clinical supplies and assembling an IND. That starts with dose range finding work, which we're in the middle of right now. So that can give you a sense of how quickly this is moving. It's going very expeditiously at this point.
Mm-hmm. Okay. And then another thing is, phase 1 trial design, the thoughts on that, if you can give a little bit color.
Sure. Yeah, so with TTR, we're talking about administering an LNP systemically. This is an IV infusion to patients. Obviously, it'll be patients, not healthy volunteers. I think that, some of this is going to be subject to regulatory discussion, which is upcoming here. But based on prior discussions with them and as we think about the field, there are patients who receive therapy today who are not benefiting from it, who don't respond. They seem like natural participants in a trial like this, given the situation that they face. The likelihood is that we would have a strong bias to neuropathy, but we're very interested in ultimately getting into the cardiomyopathy patient population, and so it's likely we'll have some of those patients as well. And the approach that we have is irrespective of whether it's a mutant form of TTR or not. So what we're trying to do is set ourselves up in a way that we'll be able to take this ultimately as broadly and generically as possible.
Okay. Maybe switching gear, we can discuss a little bit about insertion. I think you showed pretty impressive data last year. So what would be your, target insertion efficiency? What additional improvement would you try to do, and what would be your first indication? Of course, this all depends on the indication, right?
Right.
Yeah.
So as far as indications go, we've done a lot of work with alpha-1 antitrypsin. Mm-hmm. That's certainly a candidate to be the first one going forward. With Regeneron, our collaborator, we're working on factor IX for hemophilia B, and I think that's the data you're referring to. And there's other targets as well. I don't think about it as the editing efficiency as the objective. I look at as protein production. Mm-hmm. So when you're inserting a gene, typically it's to derive the benefit of whatever that gene produces. And so if it's alpha-1 antitrypsin, essentially what you're trying to do is get to levels of protein that approximate what a normal human being has. So that would be my standard. Right? Mm-hmm. Can we do that? And as I think you know, we've demonstrated that already in murine models. We'll see if we can do that in monkeys. And the same thing with factor IX. Do you produce enough of the protein to make the coagulopathy go away? And if that's 2% editing or if it's 20% editing, it doesn't matter to me. It's what does the protein do? So that's the way we keep track of this.
Mm-hmm. So do you see, current, we saw the factor IX data, right? But do you see, current what you achieve now, the editing capability is there already, or you think you need a further improvement?
Well, it was in mice, and I'm just, for the audience, the factor IX data was even super therapeutic, depending on how you did it. So you could get to levels that were tens of times higher than what's necessary to reverse the coagulopathy, and you can tune that anyway. So that's very exciting information, I think very important for patients with hemophilia B. That's in a mouse, and so moving on to monkeys is the objective now, so we can reproduce that, and then it's, a judgment for our collaborator, Regeneron, with, how to proceed.
So I know we don't have much time left to maybe discuss about the ex vivo programs.
My favorite topic.
Yeah. WT1 for AML. So wondering if you can share with us, the steps and the challenges you have for, the cell engineering.
Right. So you'll remember those 4 work streams that I laid out. The first of them is just the T-cell receptor 1, put on a regular lymphocyte in which the endogenous TCR has been knocked out. So we've done that. We've demonstrated that you can take the new TCR and put it into the endogenous locus. So all those things are in hand. What we're working through now is screening across large numbers of AML blasts from patients, those TCRs that give us the optimal effect. because ultimately what we want to have is a lymphocyte that's going to give the broadest activity for the largest number of patients. So it's a selection process right now that we're going through. And once we have that in hand, there's a regulatory interaction that's necessary for kind of a final step there. I think we'll be in a position to have what we need, and then it's moving on to making it in sufficient quantities and onto the clinic.
So what are the factors you're looking for the first clinical candidate?
It's really that broad-based killing. I mean, that's we want to have essentially an ablation of the endogenous T-cell receptor. We have that in hand. We want to have the ability to drop in our chosen TCR into the endogenous TCR site. We have that in hand. Now, the question is, of the various TCRs that we have that come from our collaborator in Milan, which one or two are going to be those best TCRs to give us the most effective, most efficient killing? So that's laboratory work that's going on even as we speak.
Okay. And lastly, just quickly touch upon the patent IP. First wanted to say congratulations. I know the 859 patent finally will be issued in the coming weeks. So, what kind of impact does this have also on your IP strategy going forward?
Yeah, I mean, it's the short answer is there's almost no circumstance where we can't do everything we need to do, and so that's very good news, and we've been confident of that for some time now. But what's taking place is last year there was a question that was addressed in the appeals court about whether or not eukaryotic cells are separately patentable or not. And we know what the answer is, which is totally different from who invented what, right? And so that opened up a logjam of a pile of patents that the University of California had waiting there to be issued. And we're starting to see that flow. And so the patent you're referring to, and one that was just issued, really speaks to the use of a single guide, which is what makes CRISPR so exciting, in any system, in vitro, in vivo, any cell type. And this is what we've been talking about for a couple of years now, that as that would issue, we would be in a very, very strong position, and that's what's happening.
Thank you very much, John.
Yeah, thank you. I appreciate it.
We will have a breakout session across the hall.